A good day to everyone. So for this video lecture, I'm going to talk about the linkage and recombination. So this is a new chapter. We are finished dun sa third chapter. So we are now on the fourth chapter. So for this chapter, I'm going to talk about the definition of linkage, determination of linkage, yung chromosome mapping na tinatawag, factors that affect the recombination frequencies, sex determination, and then sex linkage. So without further ado, let's go with the first. So what is linkage? So ano nga ba yung gene linkage na tinatawag? So yung genetic linkage can be defined as the tendency for alleles close together on the same chromosome to be transmitted together as an intact unit through meiosis. So sinasabi daw na like for example, ito yung chromosome mo. Now, ito yung allele mo. Pwede ito yung allele mo, gene 1 at saka gene 2. Now, yung linkage na tinatawag, itong dalawang ito, magkasama yan. Intact pa rin sila through meiosis. Hindi sila sumusunod doon sa tinatawag doon sa law of doon sa law ni Mendel, yung independent assortment. So walang crossover na nagaganap. Meaning itong dalawang genes na to na matatagpuan sa iisang chromosome, intact pa rin sila through meiosis. So yun yung genetic linkage. Another one is that genes that are sufficiently close together on a chromosome will tend to stick together. And the alleles of those genes that are together on a chromosome will tend to be inherited as a pair more often than not. So, sinasabi na yung mga alleles, yung mga genes na to, itong gene 1 at saka gene 2 na to, pag close to each other sila, they tend to stick together. So, nagsasama yan. Meaning, pwedeng ma-inherit mo itong dalawang genes na to na nasa isang chromosome. Yun yung mangyayari. Kasi, they are linked with each other. They are stick together nga. So, kasi they are close together. Malapit sila sa isa't isa. So, yun yung genetic linkage na tinatawag. Now, here is an illustration doon sa pwedeng mangyari sa linkage. So, dito sa isa, yun yung ina-explain ko kanina na yung link genes, yung gene 1 at saka gene 2, they are close to each other, making them likely to be inherited. Pwedeng mamana itong dalawang genes na to sa isang chromosome kasi nga, yun nga, magkalapit sila sa isa't isa. Now, yung genes on a separate chromosome naman, so kumbaga yung isang gene nasa isang chromosome, yung isang gene nasa isang chromosome, this is not considered as a linked gene. So, it is not a linked gene ito. So, and then, another example is ito. Kahit nandun sila sa iisang chromosome, if yung gene 1 at saka gene 2 are farther from each other or away from each other, it is not linked rin. So, less likely or zero chance na ma-inherit mo itong dalawang gene na to that are on the same chromosome. Kasi ito, they are not linked because they are far away from each other. Ito naman, they are not linked because yung isang gene nasa matatagpuan sa isang chromosome while yung isang gene matatagpuan sa isang chromosome. Now, this is linked gene because they are more likely to be inherited kasi they are close together. So, yun yung linkage na tinatawag. So, ano nga ba yung nangyayari kasi? So, yung normal or parang yung pag sinusunod natin yung sinasabi ni Mendel, mayroong crossing over na nagaganap. Meaning yung gene ng isang chromosome at saka yung gene ng isang chromosome, through crossover, magpapalit yan. So yung gene ng isang chromosome pupunta ngayon dun sa isang chromosome at tapos yung isang gene ng isang chromosome pupunta ngayon uh, sa kabilang chromosome. So yung crossing over can put yung mga new alleles together in a combination on the same chromosome, uh, and then kaya merong crossing na nagaganap. So when genes are far apart, tulad nitong example, di ba? Magkahiwalay yung gene A at saka gene B. So talagang far apart sila. Most likely na merong crossing over na magaganap. So yung crossing over happens often enough that lahat ng types of gametes that are produced, they will have like a 25% frequency. So yun yung sa crossing over. Doon sa pag sinusunod natin yung mga laws. So merong crossing over na magaganap. Pero doon sa linkage kasi, since they have a very short distance with each other, um, less likely na merong crossing over na magaganap during meiosis. So meaning, itong gene dito sa isang chromosome, yung gene A at saka gene B, yung dominant A at saka dominant B dito sa isang chromosome, during meiosis, walang crossing over na magaganap. So meaning, ma-inherit ito ngayon. And then ito naman, yung recessive A at saka recessive B sa isang chromosome, wala ring crossing over na magaganap. So ma-inherit rin. So yun yung linkage na tinatawag. So parang kasi short distance lang yung pagitan ng dalawang gene. So smaller yung target of crossover events. So smaller yung chances that crossing over will happen. So with that, how do we test yung linkage? So we can test yung linkage by backcrossing a double heterozygous animal to one that is homozygous recessive for both pair of genes. So ito. So you backcross itong double heterozygous, ito yung double heterozygous with that of the homozygous recessive. For both pair of gene, yung P at saka yung B. So yun, iba-backcross mo daw. So when backcrossing, ito yung pwedeng kalalabasan. So if there is no linkage tulad nito, it will follow yung law of Mendel, wherein they will segregate and then they will undergo independent assortment with each other, which will result to 4 equal phenotypes. So 4, 4, 4, saka 4. So yun yung magaganap. Pero with linkage, hindi niya fina-follow yung law of Mendel. So walang 4 equal phenotypes na magaganap, meaning with linkage na may crossing over, ito yung kalalabasan na phenotype lang. Polled black tsaka horned red. So walang polled red, walang horned black. So yun yung kalalabasan. Tapos with crossing over, kung mayroon mang maganap na crossing over, tapos may linkage, then ito yung kalalabasan na phenotype. 45 na polled black, 5 na polled red, 5 na horned black, and then 45 na horned red. So, meaning, if you cross yung homozygous recessive with that yung heterozygous, pag ito yung nakita mong phenotype niya, pag equal yung phenotype niya, then masasabi mo na walang linkage na naganap. Pero pag ito yung na-observe mo at saka ito, then there will be linkage. So masasabi mo na may linkage. So yun yung pagtetest dun sa linkage, if meron nga ba or wala. Then there are types of linkage. So, you have yung first, yung coupling linkage na tinatawag. So, coupling is the linkage of two dominant alleles of two genes in one chromosome and two recessive alleles of two genes in the other homologous chromosome. So, here yung dominant alleles of the genes are present in one chromosome while the recessive alleles are present in the other chromosome. So, sa coupling chromosome, magkasama ngayon yung dominant allele at saka magkasama yung recessive allele sa isang chromosome. So, yung isang chromosome, nandun yung dominant allele na A and B. So, yun yung coupling linkage. And then, pwede rin na yung sa isang chromosome, magkasama ngayon yung recessive A at saka recessive B. So, yun yung coupling linkage na tinatawag. Yung dominant, they are present in one chromosome, while yung mga recessive allele nila, they are present in the other chromosome. And then, another one is yung repulsion linkage na tinatawag. Now, in repulsion naman, yung dominant alleles or yung recessive alleles come from different parents and they tend to remain separate. So, here, one parental chromosome carries one dominant and then one recessive allele, while the other chromosome carries the other two alleles, yung dominant at saka yung recessive. So, yun yung repulsion linkage. So, hindi magkasama ngayon yung dominant allele sa isang chromosome. So, hindi sila magkasama, they remain separate with each other. Hindi tulad dun sa coupling linkage. So, dito sa first, sa first na chromosome, magkasama ngayon yung dominant A at saka yung recessive B. Then, dun sa other chromosome, magkasama ngayon yung recessive A at saka yung dominant B. So, yun yung types of linkages, yung coupling at saka repulsion linkage na tinatawag. Then, what is nga ba yung importance ng isang genetic linkage? So, linkage will help to keep yung parental features together by reducing the probability of gene recombination. Since naglilink na sila with each other, kung yung genes ng tatay are linked with each other, then it can parang preserve yung parental feature ng tatay. So, as a result, it aids the organism in maintaining parental, racial, and other characteristics. Kasi nga, they are linked with each other. Na ipapasa, walang recombination na magaganap. Walang, hindi nagpapalit yung genes. So, yung gene ng tatay, if they are linked with each other, yung genes ng tatay, if they are linked with each other, then, ma-preserve ngayon yung gene na yun. So, it helps yung organism para ma-maintain niya yung parental characteristic niya or pwede rin yung racial characteristic niya. So, yun yung importance ng linkage. And then, the other one, it is yung genetic linkage analysis. It will help determine whether the disease phenotype is only caused by mutation in a single gene or mutations in other genes, which can give rise to an identical or similar phenotype. So, yung genetic linkage daw, it can help determine yung disease. So, pwede kasi, since they are linked with each other, then nakita nila na with this gene, nagkaroon ng disease, then they can be able to determine kung anong gene yung nagkukos nun. So, yun yung importance ng linkage. Then, with that, let's go naman dun sa gene or chromosome mapping na tinatawag. So, gene mapping is the process of establishing the locations of genes on the chromosome. So, yun yung ginagawa nila sa gene mapping. So, nilolocate nila within the chromosome kung saan mismo yung mga genes na yon, kung saan matatagpuan yung mga genes na yon. So, nagagawa nila ito by following yung inheritance pattern uh, na de-determine nila yung position ng gene. So, before we go into yung gene or chromosome mapping, ano nga ba yung recombination? Kailangan nating malaman yung recombination frequency na tinatawag. So, when we say recombination frequency, it is a number that describes the proportion of recombinant offspring produced in a genetic cross between two organisms. So, ito yung parang frequency ng mga recombinant that is na-produce when you cross yung isang organism with another organism. So, yung recombination, it typically occurs during meiosis in which yung pieces ng mga DNA are broken down and recombined to produce new combinations of alleles. So, yun yung recombination na tinatawag. And then, yung recombination frequency, ito na ngayon yung proportion ng mga recombinant, yung mga offspring that have produces combination of allele, that have new combination of alleles, yung proportion or number ng mga recombinant na yun that is produced in a cross of the two organism. Yun yung recombination frequency na tinatawag. So here we have yung example para matignan natin yung recombinant frequency, recombination frequency. So, you have a cross between ito, yung heterozygous A at saka B, with that of the test cross or yung homozygous recessive. So, yung gametes na pwede natin makuha with this parent, ito, homozygous dominant, then heterozygous, and then homozygous recessive. While yung gametes na makukuha natin dito sa isang parent is homozygous recessive lang. So since homozygous recessive rin itong parent natin, yung gamete lang is AB, small letter AB. So yung predicted offspring out of 100 is 25 na heterozygous, then 25 na homozygous recessive which are similar to that of the parents. Kaya tinatawag itong parental. So, pareho sila, no? Heterozygous A, heterozygous B, pareho ng parent. Then, homozygous recessive A at saka homozygous recessive B, pareho ng isang parent. So, these are called parental. Now, dito sa cross na to, meron ka rin maproproduce ngayon na tinatawag nating recombinant. So, hindi parehas dun sa parent. Hindi magkaparehas yung genes with or genotype with that of the parents. So, you have here yung heterozygous A and then homozygous B. And then you have yung homozygous A at saka yung heterozygous na B. So, this is called yung recombinant natin, which are yung mga genotypes that are produced as a result of yung crossing over during recombination. So, nakakapag-produce na tayo nitong recombinant. So, meron kang maproproduce ngayon na 25 plus 25. So, para makuha natin yung recombinant frequency, yung number of recombinants which is 25 plus 25, 50, divided by the total offspring nila which is yung 100, that is equal ngayon to 50%. So, yun na yung recombinant frequency na tinatawag. So, with that, paano nga ba nila minamap yung ano natin? Yung genes natin doon sa chromosome. So, let's take for example ito. Meron siyang frequency of recombination na 17%. Now, itong kahabaan nitong isang chromosome, it represent 50 na map units. So, nasa 50%, mga ganun. So, you can map it na yung 17%, pwedeng magkalapit, pwedeng ganito kalapit yung 17% na frequency of recombination. So kung yung genes mo, yung genes, merong frequency of recombination, nakita mo na 17%, so you can map this doon sa chromosome mo. So pag naman 10%, so 10% yung frequency of recombination yung nakita mo na genes. So, medyo mas malapit sila with each other compared doon sa 17%. Then, if 39% naman yung nakita mong frequency of recombination, then meaning magkalayo yung dalawang um, genes or yung dalawang allele within that chromosome kasi nga mas mataas yung frequency of recombination. So yun yung parang pattern. So kung mas maliit yung percentage ng frequency of recombination, meaning mas magkalapit ngayon yung um, dalawang gene doon sa iisang chromosome. Kung mas malaki naman yung percentage ng frequency of recombination, um, meaning yung dalawang gene na yon magkalayo from each other, mas malayo sila, mas malayo yung distance nila within that chromosome. Paano naman if 50%? So, if it is 50%, then dito you can say na there is independent assortment na naganap. So, meaning the other gene na will be found on the other chromosome. So, kasi 'di ba sinabi ko kanina na 50% lang ito. Itong 50% map unit lang or 50% lang yung parang sabihin nating distance ng isang chromosome. So kung may frequency recombination ka na 50%, yung isang gene pumunta na ngayon doon sa isang chromosome. So hindi mo na siya matatagpuan uh, dito sa isang chromosome. Lumipat na siya. So yun yung importance ng frequency of recombination. Basta yung laging tatandaan lang is pag maliit yung frequency of recombination, pag maliit yung percentage, mas magkalapit ngayon yung dalawang gene. Pag naman malayo or malaki yung percentage ng frequency of recombination, meaning yung distance sila with each other, yung gene is malayo or kung nasa 50% na, nandun na yung isang gene, nandoon na sa other chromosome. So yun yung yung mga rules natin. So with that, let's take into example itong genes na to at saka yung recombination frequency nila. So you have yung genes na VG at saka B. Ito is yung genes on yung sa Drosophila or sa fly. Yung VG stands for yung variegated na wings. Tsaka yung B is yung black color. Then, you also have yung genes na VG at saka yung CN, yun yung color ng eye. At then, you also have yung CN at saka B. Then, nakita nila na ito yung recombination frequency. So, na-determine nila yung recombination frequency with nitong mga genes na to. So, nakita nila na yung VG tapos B na gene may 17%, yung VG at saka yung CN na gene may 8%, and then yung CN at saka yung B na gene may 9%. Now, in order to map, so in figuring out yung gene map, you first start on the highest frequency of recombination, which is yung VG tapos B na genes. So, ilagay mo dito yung VG. So, you map yung VG tapos yung B na gene. So, 17% frequency. So, pwedeng ganito sila kalayo. So, ito yung VG, ito yung B. So, ganito sila kalayo. Now, paano naman yung VG at saka yung CN? So, yung VG at saka yung CN mayroong recombination frequency na 8%. So, maaring pwedeng dalawa yung position ngayon ng CN. Pwedeng magkalapit sila dito sa part na to, dun sa left side ng VG, or pwedeng nasa right side siya ng VG. So, ganon yung distance nila. So, paano natin madedetermine kung saan nga ba talaga yung pwede nating ilagay itong CN na gene? So, tignan mo doon sa recombination frequency nila ng CN at saka B, nasa 9%. Kung ito yung ilalagay natin na gene, ito yung ilalagay natin na CN, tapos yung recombination frequency nila ng B is 9% lang, nandito yung B. So, meaning, medyo malayo. So, hindi ito 9%. Kasi they are far apart. So the lower the percentage of recombination frequency, dapat magkalapit yung gene na yon. So meaning hindi dito yung paglalagyan natin nitong CN gene. So dapat dito sa part na to. Since 9% magkalapit sila with the B gene. So yun, ganon yung pagmamap doon sa mga genes natin. So, you have here yung VG tapos B na gene, 17% yung recombination frequency. And then yung VG at saka yung CN nasa 8%. And then yung CN at saka yung B nasa 9%. So, pwedeng ganun yung, or ganun yung pagmamap natin doon sa um, genes natin. Dapat alam mo yung recombination frequency. So, ganun rin. Another example, you have yung genes na AB with a recombination frequency of 30%. You have yung BC, combination frequency of 45%. BD, nasa 40%. And then AD, nasa 25%. So, again, yung map, yung highest percentage of recombination frequency, which is yung BC. So, ganito sila kalayo. Bakit ganito sila kalayo? Remember na yung chromosome natin, nasa 50 map units, nasa 50% itong chromosome natin, yung distance nila. So, ganito kalayo, pwede yung 45%. Now, yung ano natin, um, yung B at saka yung D natin nasa 40%. So, medyo mas malapit sila when compared dun sa B at saka C. So, mas malapit ngayon yung B at saka D with each other. So, nandito na part na to. Then, so for the, ano naman, genes na AB at saka AD, medyo namali konti dito yung illustration. Medyo mas malapit sana dito sa part sana yung A, para yung genes na AD 25% and then AB 30%. So dapat dito kasi sa illustration mas malapit yung genes na AB kaysa dun sa AD. Dito sa recombination frequency makikita natin na mas dapat mas farther away yung genes na AB kasi 30% mas mataas compared dun sa 25%. So, meaning, medyo nandito dapat yung yung gene A. But other than that, ganito yung pagmamap dun sa natin, yung gene chromosome mapping na tinatawag. Then, there are factors that will affect yung recombination frequency. So, first, yung sinasabi ko is yung distance between genes. So, kung mas malapit yung genes with each other, the lesser is the recombination frequency. So, yun yung laging tatandaan. So, kung mas malapit yung genes with each other, lesser yung gene frequency. So, pag mas malayo naman yung genes with each other, kung mas malayo sila, yung distance nila, then the recombination frequency is larger, mas mataas yung percentage ng recombination frequency. So, yun yung distance between genes. Sa sex naman, meron yung um, mga species like this Drosophila. Itong heterogametic sex will show relatively lower recombination yung mga males natin. Itong mga males natin, mas mababa yung recombination frequencies nila compared doon dito sa homogametic sex natin na females. So, itong mga Drosophila na males, they present yung example because they do not show any recombination between mga linked genes. So, yun yung sex. Kumbaga may mga species, may mga insects that yung recombination frequency is yung nagdi-dictate sa kanila, nagdi-dictate doon, yung nag-a-affect doon sa recombination frequency is yung sex. Then for the age, sinasabi na yung recombination frequency as the species or as the organism grows older, the recombination frequency will go down. It will progressively decline with age. So, pataas ang pataas yung age, pababa ng pababa naman yung recombination frequency. Now, for temperature, it can affect yung recombination frequency as dun sa mga Drosophila, mababa yung recombination frequency nila kung umabot sa nasa 22 degrees Celsius yung temperature. And then for chemicals naman, may mga chemicals like yung antinomycin D na tinatawag. Yung antinomycin D, it can promote recombination. So, ini-increase niya yung recombination frequency. Then, for radiation, so radiation like yung mga X-ray natin, mga gamma rays natin, it increases yung recombination rin. So, yun. Yung pag-expose dun sa organism ng mga X-ray, gamma rays, this will increase yung recombination frequency. And then, distance from the centromere, so, yung distance niya, kung mas malapit ngayon yung mga genes, it tends to suppress recombination. Compared dun sa mga genes na malayo dun sa centromere, mas mataas yung recombination frequency nila. Kasi mas malapit, sinosuppress niya or pine-prevent yung recombination. So, yun yung mga factors that affect yung recombination frequency. Then, let's go naman doon sa sex determination na tinatawag. So, sa sex determination, it is the process of sex differentiation by which whether a particular individual will develop into male or female sex. So yun, may mga mechanisms na nagdi-dictate kung paano natin i-determine ngayon yung sex ng individual, ng organism. So, yung sex expression kasi, it is governed by yung chromosomes natin at saka yung genes. So, in mga unisexual animals, yung chromosomes are of two types. So, you have yung autosomes, so laging tatandaan nito, you have yung autosomes at saka yung allosomes. So, yung autosomes, yun yung mga chromosomes which do not differ in morphology and number in male and female. So, walang difference. Either doon sa morphology at saka yung number doon sa male and female. So, walang difference. Yun yung autosome. Yun yung chromosome na walang difference in morphology and number. Yun yung autosome. Pag naman yung chromosome na it differs in morphology and number in male and female, and then it contains genes that determine sex, yun yung allosome or sex chromosome. So, yun, laging tatandaan yun, yung pagkaiba ng autosome at saka nung allosome. So, basta pag allosome, ito na yung sex chromosome. Merong pagkakaiba, may difference in the morphology and the number in male and female. And yung And then sa allosome rin, it contains yung gene that determines yung sex. Kaya tinawag siyang sex chromosome. So ito yung nakita ko lang pagkakaiba nila. So sa autosomes refers to other than sex chromosome. So yung mga chromosomes other than yung sex chromosome, yun na yung autosome na tinatawag. So ito yung chromosomes that do not determine the sex of the individual. Autosome. Pag naman allosomes or sex chromosomes, these are sex chromosomes. Chromosomes that determine the sex of the individual. So yun yung unang pagkakiba. Autosome do not determine the sex of an individual. Allosomes, this determines the sex of the individual. Then chromosome number, autosome 2n-1, chromosome nasa 2. Then morphology is similar in male and female, while yung allosome naman, yung morphology niya is different in male and female. Then autosome, the number of autosome differs from species to species, meaning magkaiba yung number ng autosome ng isang baboy doon sa isang chicken, magkaiba yung autosome number um, ng isang buffalo doon sa isang baboy. So yun. Then allosomes or sex chromosome naman, each diploid organism, lahat daw ng diploid organism usually has 2 allosome. So yun. And then lastly, sa autosomes, it does not exhibit sex linkage, while sa allosomes or sex chromosome, it exhibit sex linkage. So yun yung pagkakaiba ng autosomes with that of allosomes or sex chromosomes. So, ano nga ba yung mga mechanism of sex determination? So first, yung sex character, then yung chromosomal sex determination, then you have yung monogenic sex determination, and then last is yung environmental sex determination na tinatawag. So first, let's discuss yung sex character. So, yung pagdedetermine natin ng sex ng isang organism using yung sex character, yung primary na inaano natin is yung gametes. So ito yung mga sperm cell at saka yung egg cell. So meaning, yung kung may sperm cell ka, then you are a male. Then if you have egg cell naman, then you are a female. Yun yung primary na tinitignan when it comes to sex character, yung mga gametes, para malaman natin kung male or female. Then for its secondary naman, yung mga genitalia. Yung genitalia. So, you have yung male, of course, you have yung penis, then yung female, you have yung vagina, yung mga uterus, mga ganun. So, yun yung pagkakaiba. So, sa male, you have yung penis, yung testicle. Sa female, yung vagina, cervix, mga ganun. So, yun yung una, yung sex character, yung tinitignan. Which is yung primary na tinitignan is yung mga gametes, yung egg cell, at saka yung sperm cell. Then, yung secondary naman is yung genitalia. Then, yung sa chromosomal sex determination naman, either by sex determination by allosomes or by diploid haploid system of sex determination. So, first let's discuss yung sa sex determination by allosomes. So, yung sa sex determination by allosome, ito yon. So, yung allosomes or yung sex chromosome are generally of X and Y type. So, yun yung parang yun yung symbol for yung male and female, lalong-lalo na dun dito sa human, so X and Y. So pag mayroon kang chromosome na X at saka Y, then you are a male, and then if you have a chromosome na XY, then you are a female. So ganon. But in some cases, like dun sa mga poultries, mga birds natin, yung chromosome nila, they are of yung Z and W types. So, yung sex with similar sex chromosome, yung XX, kung ba yung XX, yun yung tinatawag na homogametic sex. So, yun yung laging tatandaan. Kung yung chromosome nila is magkapareho, tulad ng XX, ganyan, then it is called as homogametic sex. Now, if yung sex naman nila is dissimilar, yung type, yung sex chromosome is dissimilar, like XY, yun yung heterogametic sex. So, yun yung laging tatandaan. Homogametic sex, magkapareho yung sex chromosome. Heterogametic sex, magkaiba yung sex chromosome. So, ito. There are four different systems of sex determination. So, first is yung XXXY female-male system. Then, you have yung XXXO female-male system. Then, you have yung XOXX female-male system. And then, you have yung ZW ZZ female-male system. So, yun yung apat na different system on sex determination. So, first, let's discuss yung XXXY female-male system. So, itong system na to, itong XXXY, it is mostly on mga Drosophila, tayong mga human, and then some other mammals. So, ito yung parang sinusunod natin na system, XXXY. So, in this system, female has 2X chromosome. So, yun, laging tatandaan. Pag yung chromosome is XX, XX, homogametic, then yun yung female. So, it is a homogametic and then produces only one type of gamete, which is yung X. So, yun. Then, the male naman has one X and then one Y chromosome. So, meaning it is a heterogametic and produces two types of gametes, yung X at saka yung Y. So, yun. And then, union of X ovum with yung X sperm, it will lead to the development of female, yung female sex. And if ovum unites with Y sperm, if yung X ovum unites with Y sperm, it produces yung male XY sex. So, yun. So, yun yung unang female sex determination system, yung XXXY. So, ito yung ginagamit natin sa human, dun sa mga fruit fly, and then some other mammals. So, yun yung laging tatandaan lang dito sa system na to. Pag dalawang X chromosome, then that is a female. So, dalawa yung X chromosome, meaning this is magkaparehas, meaning this is homogametic. Then, yung naproproduce lang na gamete is X. Doon naman sa male, dalawang magkaiba. Doon sa female, dalawang magkapareho. Dito sa male, dalawang magkaibang chromosome, yung X at saka yung Y. So, dahil dalawang magkaiba, then it is a heterogametic that produces two types, yung gamit na X and then yung Y. So, yun. So, paano na ganon yung pagdetermine nung sex sa XXXY system. So, paano nga ba na bubuo yung female? So, pag nag-unite yung X na ovum at saka yung X na sperm, it will lead to the development of yung XX, which is yung female sex natin. Then, pag nag-unite naman yung X ovum at saka yung Y sperm, dito, maproproduce mo yung XY, which is yun yung male sex. So, yun yung unang system of sex determination. Then, the second is yung XXXO system. So, these are for the insects naman. Yung mga grasshoppers, and other orthoptera, at saka hemiptera na mga insects. Ito yung sinusunod nilang system. So, yung XXXO na system. So, in this system, yung female has double X chromosome, while yung male has single X chromosome lang, XO. So, yung O, it connotes na wala. So, yun yung system na sinusunod nila dito. Inulit ko, yung female, pareho sila nung sa una, sa ating mga human, merong dalawang X, double X chromosome, which pertains to the female. Pero yung male, isa lang yung X, single X chromosome. Then, wala nang kaakibat na ibang chromosome. So, isang single X chromosome lang. So, yun yung pag-determine mo doon sa male or female sa mga grasshopper and other insects. So, Meaning, since yung female has double X chromosome, then it is a homogametic and then produces all the eggs with X chromosome. Then the male is heterogametic, which produces sperms, half of which have X chromosome and the other half have none. Since XO, yung O, it connotes to wala, none. So, yun yung naproproduce. Yung egg cell na napoproduce ng female, yun, dito makakapag-produce ka ng X chromosome. Doon sa male naman, yung sperm, half of it which have X chromosome, then the other half is, yun yung none, walang chromosome. Then, pag yung union of egg with sperm having X chromosome will give rise to female sex, and with sperm having none result in development of male sex. So meaning, pag nag-unite yung egg, di ba yung egg natin dito sa system na to, meron X chromosome, nag-unite ngayon doon sa sperm na may X chromosome, then yung maproproduce ngayon is female. Kasi dalawang X, double X, so female. Pero pag yung egg ngayon that contains yung X chromosome, nag-unite doon sa sperm na walang chromosome, having none chromosome, then it result in the development of male sex. Kasi iisa lang yung chromosome. So, yun yung second na system for sex determination doon sa XXXO. Then, for the third naman, this is yung XOXX system. Which yung sumusunod dito sa system na to is yung mga very few species. Medyo rare itong system na to. Mababa lang yung mga species that sumusunod dito, particularly on insects. Yung example is yung Phomia insect. So in this system, yung female has only one X chromosome and hence it is heterogametic. As a result of meiosis, 50% of eggs of such female carry an X chromosome and remaining 50% have none. So parang kabaliktaran lang ito nung second system natin. Na doon sa second system natin, yung XO, yung iisa lang yung X chromosome, yun yung male. Dito sa XOXX na system, dito sa third system natin, kabaliktaran lang. Yung XO naman, yung isa lang yung X chromosome, yun yung female. Ngayon, yung naproproduce niya lang is 50% of dun sa egg can carry yung X chromosome and then yung remaining naman is none. Kasi nga XO. Isa lang yung X chromosome. On the other hand, yung male naman, siya na ngayon yung may dalawang chromosome. Yung dalawang X chromosome. And then, it produces all the sperms with one X chromosome. Thus, male sex is homogametic. So, pag yung union of yung X sperm with yung ovum having X chromosome, it will give rise to the male sex. Tapos, yung union naman ng X sperm with yung ovum having no chromosome, none at all, it will lead to the development of female. So, yun yung third system natin. Parang magkapareho lang sila with that of the second system natin. Yung kaibahan lang is nagbaliktad. So, dito sa third system yung XO, yung isa lang yung X chromosome, yung female. Then yung double X naman, yung dalawa yung X chromosome, yun yung male. So, yung union nila, common sense na lang, pag yung X sperm nag-unite doon sa ovum or egg cell na merong X chromosome din, XX yung kalalabasan, then it is a male. Pag naman nag-unite yung X sperm with that ovum na walang chromosome din, yung kalalabasan is XO, which is yun na yung female. So, yan yung third system of sex determination. Then, yung second is yung ZWZZ system. So, ito namang ZWZZ na system, ito na yung mga birds, butterflies, and moths. So, sila yung sumusunod dito sa system na to. So, Yung female, ngayon is heterogametic and produces two types of gamete, yung Z and W types. So, dito, siya na yung heterogametic. So, siya na yung may dalawang type ng gamit, yung Z and W, yun yung female. 'Di ba? Kung matatandaan doon sa first system natin sa mga humans, yung heterogametic is yung male, yung XY. Pero dito sa system na to, yung ZWZZ, yung heterogametic na ZW type is yun yung female. Then yung male naman, it is yun na yung homogametic, and it produces all the sperms of the same type carrying one Z chromosome. So, siya na ngayon yung may chromosome na homogametic. Yung iisa lang yung type ng gamit, yung Z chromosome. So, with that, yung pag-unite ng union ng Z sperm with ovum having yung Z chromosome, it will give rise to the male. Kasi yung kalalabasan ngayon is Z, homogametic, which is yun yung male. And pag yung union naman ng Z sperm with yung ovum carrying yung W chromosome, it will lead to the development of female sex. Bakit? Kasi Z at saka W, yung kalalabasan ngayon, so yung ZW natin, it pertains to the female. So yun, yung last system dun sa ano natin, yung ZWZZ na system. With that, ano naman yung diploid-haploid na system na tinatawag? So yung diploid-haploid na system, ito, ang sumusunod dito sa system na to ay yung mga honeybees natin, yung mga ants, at saka yung mga termites. So, sa ano na ito, yung for example, yung honeybee, yung female natin have diploid. So, nasa 32 yung chromosome. And then, yung drones natin, which is yung male, have haploid. So, 16 chromosome. So, yun yung isa. Ito yung diploid haploid system na tinatawag. So, madali lang maintindihan. Pag diploid yung female, pag diploid, then it pertains to the female. Pag haploid, then it pertains to the chromosome. So, yun. So, yung 32 na chromosome, pag nakitang 32 chromosome yung honeybee, then you can say that it is a female. Pag nakita naman na 16 chromosome yung isang honeybee, then you can determine that it is a male. So, yun. Yun yung diploid haploid system. Then, yung monogenic naman na sex determination, it is the expression of sex which is influenced by a single gene only. So, isang gene lang yung nagpapaano ng expression ng sex. So, in Drosophila, yung transformer gene, which connotes ito, TRA, itong transformer gene na to, it is present doon sa autosomes that plays an important role in sex expression. So, hindi ito yung nasa allosome na ano, 'di ba sa allosome yun yung sex chromosome. So, itong transformer gene, it is present doon sa autosome. So, pero meron siyang important role in sex expression. So, itong TRA or itong transformer gene, it is recessive. Recessive siya and hence does not have any effect in heterozygous condition. So meaning, kung yung gene na yon na partner doon sa gene na dominant, then hindi magte-take into effect itong transformer gene na ito kasi nga recessive siya. Pero in heterozygous condition yon. Pero in homozygous condition, pag parehong homozygous recessive yung yung transformer gene, pag pareho silang homozygous recessive, then this gene transform the normal diploid females into sterile males. So yun yung monogenic sex determination. yung gene na ito, itong transformer gene na ito, which is iisa lang, mono lang, iisa lang na gene, it influence yung expression ng sex natin. Na kumbaga, kung yung insect na yun, yung Drosophila na yun, meron siya nitong recessive na transformer gene, recessive, homozygous recessive, yung transformer gene, then, natra-transform ngayon na from pagiging diploid female into a sterile male. So, yun yung monogenic sex determination. Then, for the environmental sex determination naman, may effect yung environment doon sa sex, doon sa sex ng species, doon sa expression ng sex ng species. So, first is yung sex determination in some organisms such as yung seaworm and horsetail plant is governed by environmental conditions and also includes some hormonal effects. So, hindi lang sa environment mismo, nandun na rin doon sa hormones nung mga organisms. So, like for example, itong seaworm, yung Bunelia, yung larva which remain free in the seawater and settle on the sea bottom are differentiated into females, while those larvae which settles on the proboscis of female develop into tiny males. So meaning, yung Bunelia daw na seaworm, pag nag-hatch na yon into larva. So, yung mga larva na hindi naka-attach doon sa female, doon sa nanay nila, kung hindi siya naka-attach doon, tapos nandun lang sa baba, nandun lang sa tubig, nandun, nag-settle down lang doon sa sea bottom, then, madedetermine ngayon, magiging female siya. So, yun. Pero yung mga larva ng Bunelia na naka-attach doon sa female, hindi siya free-flowing, hindi siya nag-float doon sa sea, naka-attach siya doon sa female, doon sa nanay, then magiging male siya. So, yun yung isang example ng environmental sex determination. Then, another one is doon sa incubation temperature. So, doon sa mga turtles, doon sa mga alligators, mga crocodiles, yung incubation temperature of egg, meron siyang effect doon sa sex. So, pag yung incubation temperature ng itlog ng mga turtle, alligators, crocodiles, pag yung incubation temperature niya is nasa 30 to 35 degrees Celsius, yung kalalabasan ngayon ng egg, nung kalalabasan ngayon ng organism na yon, yung animal na yon is female. Pero pag yung incubation temperature niya is nasa 23 to 28 degrees Celsius, yung kalalabasan ngayon ng mga animals is male. So, yun yung environmental sex determination. Merong effect yung environment doon sa pag-determine natin ng sex, doon sa pag-express ng sex ng mga organisms. Then, lastly is yung sex linkage. So, itong sex linkage lang, it is a gene that is linked to or on a sex chromosome. So, meaning, yung gene na yun, may specific na sex chromosome lang siya na nalilink na parang doon siya kumakapit. So, first we have yung X-link. So, the gene is physically located on the X chromosome lang. So, doon lang siya, yung gene na yon sa X chromosome mo lang matatagpuan. So, yung mga white eye mutant in Drosophila, melanogaster, hemophilia, color blindness in humans, these are all yung mga kalalabasan ngayon ng X-linked gene, X-linked na gene natin. So, yung gene that, yung mga genes that dictate yung white eye mutant doon sa fruit fly, yung color blindness in humans, itong mga genes na to, natatagpuan, it is located doon sa X chromosome lang. Kaya tinawag na X-link. Now, yung Y-link naman, it is transported only from father to son. So, yun. Yun yung Y-link. So examples are web toes, then yung hypertrichosis, trichosis of the ear, yung web toe daw at saka yung hypertrichosis of the ear. This yung gene that dictates these conditions, they are only linked to the Y chromosome, meaning it is only transported from father to son. Kasi yung male at saka yung male lang yung may Y chromosome, di ba? So, itong web toes, nakikita na yung mga sons have web toes, pero yung mga daughters are unaffected. So, yun. Yun yung sex linkage na tinatawag. Yung mga genes, mga certain genes, they are linked to specific sex chromosome, either X-link or Y-link. So, with that, that ends my discussion on the gene mapping, mga recombination, mga sex determination, sex linkage, at saka yung mga link genes, gene linkage na tinatawag. So, if wala medyo hindi naintindihan, then feel free to go back and then review ulit. You can rewind it para maano ninyo, maintindihan ninyo. So with that, thank you for listening.